The next section in the notes is an introduction to viruses and also eukaryotic parasites. And the point of this isn't to go into any real specific detail. What I really want you to take away from this for the exam is just getting a general overview of the major differences between bacteria, viruses, and eukaryotic parasites. The biggest thing that I want you to focus on for viruses, you all know that some of the worst diseases, infectious diseases that humans contract are caused by viruses, but there's a reason that viruses are incredibly difficult to treat. If you get a bacterial infection, antibiotics don't always work, but they often do. With viruses, they're, with a few exceptions, but usually with viruses, they're very, very difficult to treat with medication. And there are reasons for that. One is that viruses really aren't alive. They're not cells. They don't have cell membranes or organelles. They don't have a metabolism. They don't produce ATP. They can't really do anything for themselves. And what that means is their entire strategy is based on tricking the host cell, that means whatever cell they're infecting inside of you, into doing all the work for them. And that means that they must be intracellular parasites, meaning they live inside your cells, not just within your body. Bacteria can infect your blood, your extracellular fluids, live on the surface of your skin, because they make their own ATP. So as long as they can get some nutrient to use, they'll be perfectly fine. Viruses don't have any of that. They have to actually get inside of your cells. And that tells you a couple really important things about the structure and function of a virus. All right, the next thing to point out is, if you look at this picture, viruses are incredibly small. That image pretty much pushes the limit of what an electron microscope can do, and these viruses are still just tiny little balls that you can barely even make out. Each one of those is an individual viral particle, and because they're so incredibly small, that means a few things. One, they're very hard to study because you can't see them very well. Two, we're never going to see a virus in this class because our light microscopes couldn't even get close to making out a ball of 100 or 1,000 of those viruses piled on top of each other, let alone one individual one. The next thing is, because they're so small, viruses are incredibly simple, and they really are only made of two or three things. Most human viruses have all three of these components. This capsid is basically just a protein shell that sort of serves as a container to full of genetic material, either DNA or RNA. And possibly wrapped around the outside of that yet is an envelope. This envelope comes from host cell membrane. Viruses can't make their own membrane, so they have to steal it from whatever host cell they're infecting. What that means is an enveloped virus, which is, again, most of the viruses that we're going to look at, all it really is a nucleic acid, piece of DNA or RNA, a capsid shell. They're often usually very rigid geometrical figures for a reason that I'll tell you in just a second, surrounded by an envelope full of membrane that was taken from the host. And those envelopes may have proteins in them. They're often called spikes that are used to sort of dock and connect to the host cell membrane. What this really means is a virus is, if you say it sort of plainly, a virus is just a container, right, a capsid in an envelope, filled with DNA that's really just instructions for how to make more capsids. So it's essentially a container filled with instructions for how to make more containers. And the only thing a virus really has to do, which is quite difficult, is convince your cells, the host cell, that it should take those instructions, the DNA or RNA, and use it to transcribe and translate into more capsid protein. And so it's essentially turning your cells into sort of unwilling virus factories. Another thing that all of this means is a really large part of a virus's effort or concern or strategy is based around trying to get inside the host cell. Because once it's inside, the DNA can take care of everything. Right? The viral DNA is very, very elaborately involved to trick the host cell into transcribing and translating it. And so the only thing left is, how do you get the DNA inside the cell? And I show you these pictures, these illustrations of viral shapes and sizes to show you that there is a lot of variety, but a lot of them have shapes and surface proteins, which you can't really see here, that very much enable them to get inside the host cell. In class, I used the example of this phage virus that contains its DNA in this open capsid space up top, uses these spikes to serve as stabilizers to land in the cell membrane, uses these spikes to puncture a hole through that membrane, and then insert that DNA into whatever the host cell may be. And so just remember, a virus doesn't do anything outside the host cell. They float around, they're inert, they don't move, they don't metabolize, they don't do anything. Their only goal is get their genetic material inside the cell, start making copies, and then spread to other cells. All right, the next section in the notes, I'm not going to spend much time on in the video because it's very straightforward. Just remember, the vast majority of diseases that humans encounter are bacterial or viral, but there are still a decent number of eukaryotic parasites. Some of them are definitely microscopic. For example, fungus, we care mostly about yeast. Yeast cause a good amount of sort of mild problems, but usually not very severe ones for humans. Protists, these are single-celled eukaryotes. Worms, they're not really microscopic, usually. They're very large, but they sort of fit along with the parasite theme in micro, so we put them here. And so for right now, don't worry about details. We're just looking at sort of general categories of eukaryotes, and you should keep those in mind going forward. The last category of infectious agents, and I will spend a little bit of time on here because they're kind of strange, are prions. And prions are characterized by one very, very unique feature. And that's that they don't have any DNA or RNA, which is really strange. So no nucleic acid. And you guys know, if something wants to replicate, it has to have DNA or RNA. Every other living organism has it. All bacteria, prokaryotes, and all eukaryotes are DNA-based. Some viruses use RNA, but either way can work. Prions are really strange in that someone noticed a disease, Kuru, that we talked about in class, where they could very carefully prove that something was being transmitted from one person to another, infecting their brain. But there was no DNA or RNA present, and that's really weird. And the model that was come up with is that it's all protein-based. And what that means is, inside a person, some proteins, this is very rare, but some proteins, and we'll call these in general prion proteins, have what's called a normal, healthy, standard shape or three-dimensional function. And this blue cube is supposed to represent the normal, healthy version of that protein. It's possible for that protein to be converted into a different shape, in a different form, that's pathogenic in some way. And what this idea is saying is this conversion from the normal version to the prion version is usually blocked because it's just too energetically unfavorable. The physics just don't work out. But something strange happens. If you expose yourself, so in the example of Kuru, this means eating the brain of someone else who had Kuru. Obviously, that's not really happening anymore because people have been educated about it. But if you are exposed somehow, or this could be eating contaminated meat from a cow with mad cow disease, that by exposing yourself to the prion form and having that prion form come in contact with the healthy version that's already inside of a person's brain, when those two touch each other, the prion protein converts the healthy one into a prion protein itself. And from there, it just keeps spreading and spreading and spreading. And so in that way, this prion protein isn't really copying itself. It's not making anything new. It's just taking existing healthy protein that would have been fine, and converting it to the prion form. And I usually liken it to knocking down dominoes. You're not making any new dominoes, you're just kicking over the ones that are already there. You're converting the healthy protein that was already in that person to an unhealthy version. The second way that this is thought to happen is a lot of neurodegenerative diseases like Alzheimer's and Parkinson's probably fit this prion category, but they're not contagious. In that case, there probably is something inside of a person that allows those proteins to spontaneously convert to the prion form and then start the spreading process. The big question that's been very, very hard to answer is, why do some people start converting to that prion form and others don't? And that's the big issue. So from this, take away from it, well, what is a prion? How does it spread? Why does it seem strange that it doesn't have DNA or RNA and can't replicate? And if you can sort of explain all of those examples, then you should be in good shape. Okay, this slide starts a section on genetics and gene regulation. And what I want you to see from this is that the DNA sequence of a microorganism, or any organism, matters a great deal. So just knowing DNA sequence is important. But what happens after that is just as important. And along the way, there are lots of different ways that activity can be regulated. What I mean by that is, you have a piece of DNA, but just because you have it doesn't mean you have to transcribe it to RNA, right? That gets regulated. Just because you now have an RNA, it doesn't mean you have to translate it, right? That gets regulated. And just because you translate a protein doesn't absolutely mean the protein has to be active and actually doing something. Right? That can be regulated too. And I'm not going to go into an enormous amount of example with this, but what I want you to see is that all of this regulation business, everything that's going on here, is based on response to the environment. Right? I'm going to use the example of the lac and trip operons later as an example of transcriptional regulation, but it happens on all the other levels too. When cells encounter a different environment, they're going to start changing which genes are transcribed and translated, which proteins are active at what time. And since protein always determines function, that's important. And so along the way, if you're going to study what is a microorganism doing, how is it dangerous, you would want to know first, what's the sequence of the gene. It's nice to know the genomic sequence of that bacterium. And second, when are those genes being active and when are they inactive? All right, quick overview. This is mostly just for, not memorizing numbers, but for just giving you a broad overview of the vast difference between human, bacterial, and viral genetics. The biggest thing is size. Viral genomes are very, very small. Thousands of nucleotides, as opposed to humans that have billions of nucleic acids in their genome. And bacteria are somewhere in the middle, in the millions category. And the only real benefit of being small is being fast. Okay? And so viruses are taking a strategy where they're incredibly small, they're incredibly fast, but they're also extremely dependent on the host cell. Right? They can't do much for themselves because their genome is so simple. Humans, on the other hand, are very large, we're incredibly slow from a reproductive standpoint, but we're very adaptive. We're really not much dependent on other organisms for survival. Okay? So part of it's just a size thing, and the trade-off is speed. The other thing that I want you to see is human genome is about 1,000 times larger than an average bacterium like E. coli, but we only have about 6 times as many genes. What that means is, if you go into a bacterial cell, 80-some% of their DNA directly codes for protein. That number is only about 2% for humans. This is entirely a matter of efficiency. Okay? There is an enormous incentive for bacteria to be efficient because, again, their entire strategy is based on speed. They can't deal with replicating extra DNA because it slows things down. And their entire goal is reproduce as fast as possible, partly to evade your immune system and partly to compete with other bacteria. Okay. So this next section is just a very quick reminder about transcription, translation, and protein sequence. What I want you to take from this is when transcription happens, right, so we've got a template strand of DNA, a coding strand of DNA, and RNA polymerase is moving down that piece of DNA, making a messenger RNA. What I want you to take from this is not the details of 5' and 3' and coding strands and template strands, but just remember, this RNA should be essentially a perfect copy. I know it's a slightly different molecule, but it's essentially a perfect copy information-wise of what was in that DNA. And there's no other purpose to making that RNA except to use it to translate a protein. And when a ribosome reads a piece of RNA, the only thing that it's doing is moving down that RNA strand and adding amino acids. These little guys coming off the ribosome are supposed to represent amino acids, making the growing protein that's being translated. Okay? And what that means is, if you know the sequence of DNA for a bacterium, you know the sequence of RNA. And if you know the sequence of RNA, you can use this table to produce a protein sequence. And so if your RNA sequence was AUG, you start with the methionine. Let's say it was AUG, AC, A, threonine, AAU, asparagine. You keep moving through, you know exactly what the protein sequence is going to be. The significance of this is that DNA sequencing has become very doable. It's getting cheap. And so when you have a bacterium and you don't know what it is and what it's doing, the thing that you can now do is just sequence its DNA. Right? You get a genomic sequence, and knowing the DNA sequence itself isn't really that big of a deal, except that it tells you what the protein sequence is going to be. And since protein determines function, that's perfect. Toxins that bacteria release, surface proteins that viruses use to get inside your cells, enzymes that give bacteria antibiotic resistance, they're all protein-based. And so if you can get a handle on what the genetics, what the DNA sequence of the pathogen is, you can use that to connect it to function and try to reason through how difficult or how pathogenic that virus is going to be when you have to deal with it. All right. To further speed up the process, because remember, this theme of replicating and dividing and responding as fast as possible is a theme that'll keep going through the class. All this is showing is, I think it's a cool picture. This is an electron micrograph of, there would be a strand of DNA up here that you can't see. That's this. A ribosome, an RNA polymerase producing an RNA. That's the strand right here, same one that's shown in red on this picture. And each of these little black balls is one ribosome moving down that RNA. And these little coils coming off the side are newly formed proteins. All that's showing you is it's easy to think that bacteria not having organelles, but they don't have a nucleus, for example, is a disadvantage. Here they're using it as an advantage because the fact that they don't have a nucleus means that transcription and translation can happen all at the same time. And in a eukaryote, this RNA would still be being made in the nucleus, have to be exported yet. In a bacterium, they're already moving, and they're producing the first batch of protein before the RNA is even done being made. It gives them a really enormous speed advantage. Okay. Very quickly with mutations, there are different ways that DNA can be mutated and changed, and this can be beneficial or it can be damaging as far as humans are concerned. In some ways, we use DNA mutations in bacteria to cause problems. For example, UV light, you'll use UV lamps sometimes to sterilize surfaces. UV light is used to sterilize food in production factories. And if you mutate enough DNA, then the cell will just die because there's too much damage. If there are smaller changes, then that can get bad because sometimes it can actually lead to new, beneficial advantages for the parasite. In general, substitutions are more mild. Deletions and insertions are usually very damaging because they cause frame shifts, and I showed you that on the translation tool in class that I use. It's kind of difficult to illustrate here. All right, next section I've got is on going back to that regulation bit that we talked about earlier. This is specifically talking about transcriptional regulation. This picture is just to give you an example of how complicated things can get. In eukaryotes, there's an enormous amount of regulation to the point that if you had a gene right here that was responsible for coding for a single protein, all of these transcription factors, each one of these blobs on here, MIC and MAX, MOF and NELF, weird acronyms, all of these transcription factors are just binding to regulatory DNA. This coil is supposed to represent that. And the goal of regulatory DNA is just to control the transcription of a gene. And so eukaryotes put an enormous amount of effort into controlling when and how and why a certain gene gets turned on or turned off. That explains at least part of this observation, right, that only 2% of a human genome codes for protein. A lot of that other 98% is regulatory. It's putting a bunch of effort into controlling when does this gene get activated. In bacteria, the same thing happens, but it's much simpler. And so as an example of that, I'm going to go through two operons. An operon is just a cluster of DNA with a regulatory sequence and then usually a handful of genes pushed together. Humans control one gene at a time. Bacteria take the efficiency strategy of sort of smushing similar genes together in a cluster so you can control them with one sort of shared regulatory sequence. And the goal of the lac operon is allow bacteria to use lactose, but only when glucose is not available. So since glucose is more abundant and it's more efficient, bacteria will choose to use glucose first, but if it's not available and there's some lactose around, you want to take advantage of that. And so what this means is these bacteria, they have the ability to use lactose, but you don't want to actually make the enzymes that break down lactose unless you have to, because otherwise that'd just be very wasteful. You'd be wasting time and energy. And so when you're not using lactose, the operon needs to be shut off. Okay? And I think this figure summarizes it fairly well. You've got four possible scenarios. In all of these, the only scenario when you actually want to turn the operon on is this one. Glucose levels are low, which means you don't have glucose available. Lactose levels are high, so you want to use it. Okay? And what that's showing is these lac genes, right, this schematic up here, LAC-Z, LAC-Y, and LAC-A, are three different genes that code for three different enzymes that are needed to use lactose as an energy source. And so when lactose needs to be used, turn on transcription. Right? That's what strongly expressed in this case means. You're turning on transcription, making a new messenger RNA. Right? In these other situations, if glucose levels are high or lactose levels are low, in all three of those, operon needs to be off. Okay? You either have enough glucose or you don't have lactose. There's no reason to make those enzymes. Operon should be off. The question in all of this is, how does that actually work? Bacteria aren't smart. They can't see that there's lactose there, and they don't know, they can't think and decide to turn the operon on and off. So there has to be some kind of sensor. Okay? The lactose sensor is this protein called a repressor, and its name sort of gives away the function. It represses expression of the genes in the lac operon. And if you notice, that repressor is stuck to the regulatory piece of DNA. Right? This part up here is all the regulatory DNA. These are the actual genes over here. Anytime lactose levels are low, the repressor is stuck to that regulatory DNA, transcription's turned off. And that makes sense. If you don't have lactose, you don't need to make those enzymes, and you want to turn transcription off. Right? When lactose is there in either of these two situations, right, you see the repressor's gone. It's not there. And that's because lactose directly binds to the repressor protein, pulls it off the lac operon. It's just like a neurotransmitter or a hormone binding to a receptor and making it change shape. Lactose binds the repressor falls off the DNA, the operon DNA, and it doesn't block transcription anymore. That's perfect. The second thing in all this is you have to figure in glucose. And this little blue blob right here, it says, ignore the CAMP. That's a sort of a second messenger that controls the whole process, but we're not going that far. You can think of this blue thing as being called the CAP protein. It stands for catabolite activated protein something or other. All it means is when glucose levels are low, that CAP protein is present. The difference is CAP protein is an activator of transcription. It turns it on. That's perfect. When glucose levels are low, you want the operon to be able to be on. And when glucose levels get high, that CAP protein's gone. You lost your activator, and the operon's off. And so in order for the operon to be on and transcribing a lot of RNA, two things have to be true. Low glucose means your activator, the CAP protein, is present. High levels of lactose means the repressor is absent. Right? You want to have your activator, get rid of your repressor, and then those genes can be turned on. And that works out perfectly because those genes, remember, code for enzymes that will break down that lactose, which is exactly what the goal of the whole thing is. The second example of this is the TRP operon. TRP stands for tryptophan. And it's the exact same idea. Tryptophan, which they've got represented in red here, binds to its repressor. The only difference is, in the lac operon, the enzymes that you're making break down lactose. And so you need to make those enzymes when lactose levels are high. In the TRP operon, those enzymes are synthesizing tryptophan. And so you need to make those enzymes when tryptophan levels are low, because that's when you need to build more. Right? Tryptophan's an amino acid. It's a building block for translation. And so everything's flipped. Instead of tryptophan pulling its repressor off like lactose does, tryptophan puts its repressor on the operon and turns transcription off. Right? Think it through. When tryptophan levels are high, you don't need to make these enzymes. Right? You already have enough tryptophan. There's no reason to make more. And so tryptophan turns its operon off. Same principle, right? Environmental conditions, amount of tryptophan, is controlling transcription. It's just the on-off is flipped compared to lactose. All right, the last little section that I'll cover in this video is some information about ATP production, aerobic and anaerobic respiration. And we're not going through a ton of molecular biochemical detail here. All I want you to remember is bacteria have to make ATP, and eukaryotes for that matter, the exact same way that humans do. ATP is the same molecule across pretty much all organisms, and the substrates are the same. Glucose, fat, protein, the same things that you use for calories are, for the most part, the same things that bacteria use for calories. That makes sense because when a bacterium infects a person, the person is their energy source. Right? They're taking those molecules from your tissues and using them for cellular respiration. In that entire process, just remember, it's all about trading electrons and protons to some degree. And so the decision, the important thing that we're going to talk about here is aerobic versus anaerobic respiration, because bacteria have the ability to be anaerobic more so than humans do, at least to live off it. And so the whole electron trading, proton passing thing becomes important, because the decision of whether or not you can be aerobic or anaerobic comes down to where are the electrons moving, where are the protons moving. And so just remember, we're not going through all the details of glycolysis and Krebs cycle and all that business, but the processes involved there really just come down to trading around electrons and protons. Okay. And this schematic, you should remember, at least vaguely from a past class. All it's showing is, if you start with glucose, and I should note, it doesn't make it into textbooks, but remember, fat and protein can be used to feed into the same process. If you start with anything that has calories, like glucose or fat, the goal is try to get it into Krebs cycle, pass off electrons through these intermediate molecules like NADH and FADH2 to an electron transport chain. That's what gets you most of your ATP at the end. The break point is right here. And in eukaryotes, that's the entrance into mitochondria. Right, that last molecule of glycolysis is pyruvate. Bacteria don't have mitochondria, but they do a very similar thing along folds in their cell membrane, their plasma membrane. And so the same thing's happening in bacteria. And if you have oxygen, remember, oxygen accepts those last electrons and protons to become water. Right? Glucose or fat plus oxygen gives you CO2 and water. If you don't have oxygen, there's nowhere for those electrons or protons to go. And the break point here is right there with pyruvate. If you have oxygen, you can keep going towards Krebs cycle and you're aerobic. If you don't have oxygen, you have to shoot off to the side and become anaerobic. Right? And that's where fermentation comes in. Okay. So, just remember, this pyruvate, that's the end product of glycolysis. Also, don't forget, glycolysis does produce some ATP. It's not much, but it's not zero. And so if bacteria are anaerobic, what it means is they can't do that. They can't go through Krebs cycle. They can't finish the electron transport chain. They can't make all that ATP that you would really like to come out at the end. It doesn't happen. And so if there's no oxygen present, what that means is the only ATP that those bacteria can get are from glycolysis. The problem is, if they keep going through glycolysis over and over and over again and keep making pyruvate, that pyruvate doesn't have anywhere to go. It can't go into Krebs cycle, and so you've got to get rid of it somehow. The two options for getting rid of it are to convert it either to lactate or ethanol. Some bacteria make lactate, some make ethanol. Yeast and other eukaryotes or fungi tend to make ethanol. It doesn't really matter which in principle. The point is, you've got to make that pyruvate go away, right? Because if not, it's just going to build up and traffic jam backward into glycolysis and prevent them from making ATP. And so this is incredibly important for humans because fermentation like this is happening inside of our gut. That's why humans are exposed to low levels of ethanol, even if you don't drink any. Obviously, we've harnessed the power of that in yeast to brew things like beer and make wine and whatnot. It also matters because studying bacteria that are anaerobic is very difficult. The majority of your GI tract is composed of anaerobic bacteria, and they're kind of difficult to study because some of them are even sensitive to oxygen to the point where it's poisonous. They can't even handle oxygen. And so pulling them out of a person's GI tract and using them in a lab is difficult because you have to keep them protected from that oxygen. Of course, if you do have oxygen, bacteria, for the most part, the ones that are capable of being aerobic will use it. And it's the same thing that happens in humans. Again, it's in their plasma membrane, not their mitochondrial membrane. But just remember that finisher. Those molecules that I pointed out earlier, NADH and FADH2, the whole goal is take their electrons, it's here, hand them off to this electron transport chain, build up that proton gradient. In bacteria, it's across the cell membrane, and eukaryotes across the mitochondrial membrane, and let those protons flow through ATP synthase to make a bunch of ATP. And so aerobic respiration is still preferred. Most bacteria will do it if they get the chance because it allows them to grow faster, but a lot of them are also, like I said, very flexible and can use anaerobic respiration. All right, last thing to point out, all of these processes are very tightly controlled. And we're going to spend some time in the next section talking about enzymes and inhibitors. And I just want to take the opportunity to point out here, this happens a lot, not just in using drugs and medications as antibiotics, but inside a normal cell. A good example of this is ATP acts as an inhibitor to enzymes in glycolysis. A common example is an enzyme called phosphofructokinase. And what I want you to see is it's just a negative feedback setup. Bacteria have to control how much ATP they make and when they make it. And so if too much ATP starts being made, it actually acts like an enzyme inhibitor to enzymes in glycolysis. That should slow down the rate of those reactions, and ATP levels should go down. If they start getting too low, this inhibition goes away, everything should speed up, and you should start making more. And so the entire thing is built based on this negative feedback where the end products, things like ATP and other things near the end like citrate, go backwards, right, negative feedback, and actually control the rate of glycolysis further back down the line.